honorable guest Dr. K.E. Lavande sir, former Vice Chancellor Dr. Balasaheb Sawant, Konkan Krishi Vidyapith, Dapoli. And also welcome to Honorable Speaker Dr. M.G. Palshetkar sir, Assistant Professor, Department of Agricultural Botany, Dr. Balasaheb Sawant, Konkan Krishi Vidyapith, Dapoli, and all those who are joined in today's seminar. I request to Dr. Sushma Kirtane ma'am, Organizing Secretary, to deliver the introduction as well as welcome address of today's webinar.
First of all, I tribute to the great personalities not only of India but the world also. Chhatrapati Shivaji Maharaj, Mahatma Jyotiba Phule, Savitribai Phule, Rajashri Shahu Maharaj, Dr. Babasaheb Ambedkar, Swargiya Yashwantrao ji Chavan Saheb, Swargiya P.D. Patil Saheb, and all who have devoted their lives for betterment of society. Honorable President, Dr. Apparao sir, Dr. K. Lavande sir, former Vice Chancellor, Dr. Balasaheb Sawant, Konkan Krishi Vidyapith, Dapoli. Honorable Speaker, Dr. Palshetkar sir, Assistant Professor, Dr. Balasaheb Sawant, Konkan Krishi Vidyapith, Dapoli. Honorable Principal, Dr. S.B. Kengar sir, Honorable Principal, Dr. S.L. Lavre sir, Dr. Poddar sir, and all my colleagues and all respected participants. I feel extremely privileged and honored to be given this opportunity to welcome you all. On behalf of our college, Yashwantrao Chavan, College of Science, Karad, and Arts, Commerce and Science College, Sonai, Ahmednagar, I welcome you all and thank you for taking the time to join us today. This online platform is very useful and effective. Participants can join from various places, localities, can share their ideas and discuss on various aspects. I think 3 C's, that is communication, collaboration, and creation, are very essential for improvement of knowledge. We have decided to celebrate 200th birth bicentennial of the great genius of genetics, father of genetics, Gregor John Mendel, by organizing series of national webinar. I tribute to Gregor John Mendel on his birth bicentennial. We salute him as a paragon of science. One important thing I want to share here, that when I was doing my PhD in Pune University, today's guest of honor, Dr. Lavande sir, has given us a permission to conduct the trials of mutation breeding program. Because of his valuable, precious help, I could successfully completed my research work at NRC for onion and garlic Rajgurunagar. Sir, I will never forget this help. Thank you very much, sir.
Thank you.
On this second webinar of the series, I warmly welcome you again and thankful to Honorable President, Honorable Guest of Honor, Honorable Speaker, dear participants, for sparing valuable time with us. Jai Hind, Jai Maharashtra, Jai Bhim. Thank you. Thank you.
Thank you, ma'am. I request to Principal Dr. Shankar Lavre sir for convener speech.
Thank you, Arangre sir. First of all, I must mention that for this particular webinar, part 2, we have with us Dr. K.E. Lavande, former Vice Chancellor, Dr. Balasaheb Sawant, Konkan Krishi Vidyapith, Dapoli, and Dr. M.G. Palshetkar, Assistant Professor, Agri Botany, Dr. Balasaheb Sawant, Konkan Krishi Vidyapith, Dapoli, as well as my guide and mentor, Dr. B.J. Apparao, Director, Collegiate Education, Mula Education Society, Sonai, all the participants and colleges who are participating in this particular seminar and helped in organizing the seminar, that is Yashwantrao Chavan College of Science, Karad, and Arts Commerce College, Sonai. So I welcome you all on behalf of these 2 colleges and all the authorities of the colleges. All the participants who are participating in this definitely get knowledge and will definitely learn many more things from this webinar because Mendel has given us factor, Mendel has given us different laws, you can say, which are useful in genetics. And now we can say these laws are also helpful for, can say, today's understanding of, can say, molecular genetics and understanding of, can say, gene and factor and DNA. I think this is a great contribution given by Mendel. Today itself, it is also useful. So on behalf of college and college management, I welcome you all. Thank you very much.
Thank you, sir. Then I request to Professor Dr. Sandeep Khedkar, sir, to introduce today's guest, Professor Dr. K.E. Lavande, sir, and Honorable Speaker, Dr. M.G. Palshetkar, sir.
Thank you, sir. I really feel honored today to introduce today's dignitaries. Firstly, Eknath Lavande, sir. He is a former Vice Chancellor of Dr. B.S. Konkan Agriculture University, Dapoli. He also acted as a former Director of National Onion and Garlic Research Center, Rajgurunagar. As long as 40 years of experience as a teacher and researcher in the Department of Horticulture. He developed 13 varieties of vegetables, 9 varieties of onion, and 3 varieties of garlic during his tenure. With his drop irrigation and storage of onions, he could increase the yield to 40 lakhs of ton from 4 lakhs. He has been honored with numbers of awards in the state and in the country. Today's speaker, Dr. N.G. Palshetkar, sir. He is an assistant professor in the Department of Botany, Dr. B.S. Konkan Agriculture University, Dapoli. He has published 25 research papers and developed 3 varieties in his tenure. He also worked as assistant professor at College of Agriculture, Dapoli, Agriculture Research Station, Palghar, and College of Agriculture, Dapoli. With this, I would also like to introduce today's president of this program, Honorable Dr. B.J. Apparao, sir. He is Director, Collegiate Education of Mula Education Society, and he has been a mentor, guide, and a source of fountain for us. So with this, I welcome all the dignitaries of this program. Thank you.
Thank you, sir. Then I request to today's guest, Professor Dr. K.E. Lavande, sir, for his valuable guidance.
Thank you, and good afternoon to all of you.
Good afternoon, sir.
Honorable members of this organization committee and the today's speaker, Dr. Palshetkar. Topic aptly selected for the today's program is heredity before Mendelian and after Mendelian. Friends, the phenomenon of heredity was same before Mendelian, and it is same after Mendel also, and it is going to be continued in the future so long the life on this planet is available as in the same fashion. However, the ways of understanding changed with the innovations, or rather scientific innovations, and with that there has been tremendous advancement in the science of genetics. The history of genetics dates back with the classical era of Pythagoras, Hippocrates, and Aristotle. The understanding of life at that time, during their era, understanding of life, plants, organisms, was in a primitive stage. And sometimes there were many funny ideas also. But the real understanding came after the Mendel's work. The Augustinian friar Gregor John Mendel, commonly known as Gregor Mendel, who was a priest in Austria, and as a hobby, he used to grow vegetables in his backyard, church backyard, and he studied the genetics and population of pea. Everybody knows the seed coat color, seed surface, and he published the report or other research papers in 1866. Until after that, almost 34 years, the work was unnoticed. It was almost hidden for 30 years. And then it was rediscovered in 1900 by three scientists, Hugo de Vries, Karl Correns, and Erich von Tassenmark. They brought the wonderful work of Mendel into the light. And Mendel was widely accepted by the scientists in 1925 with the confirmatory work of Morgan, who did similar type of work on Drosophila, the eye color and maride and all this, as these characters segregated in the same fashion that of phi, and this was confirmed. And his laws, which while working and publishing his paper, 4 laws he postulated, they were also confirmed on the basis of 2 and 4 alleles transferred from parents to their offsprings, generation after generation. And therefore, we can define heredity as the transfer of traits from one generation to other generation through sexual or asexual means. He postulated 4 principles which will be obviously discussed in detail by today's main speaker. The principle of paired factors. There are alleles and they are always in pair. The principle of dominance. One allele of them is in a dominant position, other is in recessive position. And there is a law of segregation. While sexual mating or at the time of meiosis, these alleles segregate from each other. And then 4th law he postulated is the independent assortment. They are channelized independently to form a different combination. And these were well accepted by the scientists at that time. But while understanding the complex characters, it was good that some characters or traits which have a high heredity can be explained on these phenomenon or these principles. However, the characters like yield, quality, which are governed by too many genes, microgenes, and which do not follow the Mendelian pattern, the scientific community was confused with these observations. And then later on, they started applying mathematics, statistics to that, and then came into existence population genetics, or commonly called as quantitative genetics, in the year 2025 to 250, 25 years. With that, a lot of varieties in croplands were developed through population genetics. Still, it is working in many cross-pollinated vegetables like onion and all that. The molecular genetics started in 1940 to 50. That the understanding started with that. Earlier understanding was about the organism, and the people knew that organisms are made of organs. Organs are made of tissues made of cells, and cell has nucleus. And so also the nucleus has chromosomes, and there are genes in the form of DNA and the chromosome. the cytologists, and then biologists, when all work together for understanding this phenomena, now we are in a position to know that DNA is a portion of chromosome today. And this become obvious and very clear when Watson and Crick, almost in 1953, they searched or they formulated a double helix model of DNA, and there was tremendous change in the genetics. This will be called as ultra modern science after Mendel. Now we know about gene sequencing, which we are talking about 60s and 70s about sequencing, but real sequencing came in the last 2 decades. There is genome sequencing. Whole genome is now known in many cases, like wheat and rice and potato and tomato. Even for that matter, the human genome is complete. And we were partner in sequencing one chromosome of, rather, ICR was the partner of rice. And through this, we are able to know which gene is situated on which chromosome, at what distance from the centromere and all that. And through that, advanced technology of crop breeding has came into existence since last 30 years. That is called genetic engineering. And through that, it became possible to incorporate genes, not only from species, but from across genera, and the organisms called as transgenics, where the classical example is of cotton, where the gene from Bacillus thuringiensis fungus is transferred into cotton, and that works against bollworm. There has been a tremendous revolution in all over the world in enhancing yield of the cotton, but unfortunately the yield is plateauing. There are various reasons for that, because pests are becoming resistant to this kind of technology, and many companies now they are working hard. You know, the Monsanto, whose budget is almost bigger than in a bigger country, they are investing a lot on understanding the genome and sequencing the genome so that they can develop croplands varieties. But this topic, although we have varieties in cotton, we have varieties in soybean, not in India, but in our country, we have a different view. I would rather boldly say that rather he is not pro-technology. We always opposed the transgenics, but to feed the nation in coming future, we need to have need to accept this kind of technology. With the best technology now, the country has permitted is gene editing, and whole world is looking for gene editing. Among the species, the harmful genes or the genes which are not useful for commercialization of a croplands or for a human being, for other organisms or animal which create disease or chronic disease. And now whole modern science is whirling around the gene editing technology. So this is the tremendous development in genetics over last 100 or 200 to 150 years, I would say. And the base is definitely laid down, or the foundation is laid down by the great scientist or genetist, Dr. Gregor Mendel, and the whole world will owe his efforts and will remain in debt of his and other scientists. Thank you very much, and I wish great success for this webinar. Thank you all.
Thank you very much, sir. Then I request to today's speaker, Dr. M.G. Palchetkar, sir, for his session.
Good afternoon, one and all. Today's guest of honor, respected Dr. K. Lavande, sir, ex-Vice Chancellor of my university, and all the participants over here, maybe from teachers and from all the students here. Before I start my session, firstly, I must say the thanks to the honorable organizing committee or all the organizing members for organizing such a national webinar series, and they give me that opportunity to share my knowledge on the occasion of the bicentenary birth anniversary of the Gregor Mendel, father of genetics. So today I'm going to share the knowledge regarding the pre and post Mendelian concepts of the heredity. During my speech, I will cover that whatever that pre-Mendelian concepts, the pre-Mendelian concepts in relation to that, how the characters which are transmitted from the one generation to the another generations. So number of workers worked on that, and they put forth that their ideas, their concepts, their theories regarding the transmission of the characters, how the characters transmitted from the one generation to the another generation. And another thing is that how the progeny, how the offspring, those were developed. And I will cover that pre-Mendelian concept regarding the transmission of the characters, then in short about the Mendelian work, and then lastly about the Mendelian deviations. As Dr. Lavande sir, respected Lavande sir, pointed out that the Mendel work is Mendel work. Nobody can change that. Nobody can challenge that. So that whatever that Mendelian deviations are there, that is only some differences from the Mendelian findings. So those comes under that Mendelian deviations. So this one is the first vapor theory proposed by the Pythagoras before 500 BC. According to this theory, so whatever that organisms, every part of the organisms, they are emitting some kind of the moist vapor, and that vapors from the different parts of the organisms, those are aggregate, and there is the formation of offspring by the aggregation of the vapors, those from the all parts and get aggregated to form the offspring. That is the first theory was proposed by Pythagoras. Second one, the fluid theory. This theory was put forth by Greek philosopher Empedocles. According to this theory, each body part,
Make it full screen.
and that fluid from the different body parts, which from the different parents, from the two different parents, it is mixed up, and there is the formation of the embryo. This is the second theory related with the pre-Mendelian theory. That is also pre-Mendelian era theory. So this is the second theory of that fluid theory. Third one, that is spontaneous generation theory, as I told you. Regarding that development of the living organisms could arise from the non-living matter, and that was process, whatever the development of the living organisms from the non-living matter, it was regular in nature. So that theory was called as a spontaneous generation theory, and that theory was proposed by Aristotle, a Greek philosopher. The pre-formation theory, according to this theory, and that theory was proposed by the two Dutch biologists, Van Dam and the Bonnet. And this theory, according to this theory, the miniature human body, it was already present in the egg cell and the sperm cells, and that miniature human body was called as a humunculus. So that miniature human body perform in a gametes at the time of the development of the zygote formation. So there is the growth of the miniature human body, which was already present in the egg cell and the sperm cells. So, but that theory was soon given up because of that. This could not prove the scientifically. Fifth theory was theory of epigenesis. This theory was advocated by Wolf, a German biologist. This theory states that the eggs and the sperm cell doesn't have any miniature human body.
So earlier worker stated that miniature, that is the egg cell and the sperm cell having with that of the miniature human body, and after that, at the time of the zygote formation, there is the development of the human body, there is the growth of that human body. So, but this theory of epigenesis state that the eggs or the sperm doesn't have any miniature human body. So that eggs and sperm cells are undifferentiated. So differentiation of that egg cell and the sperm cells into the various organs or the different parts, it was takes place only after the fertilizations. So there is the development of that different parts, differentiation of the various parts of the various organs only after the fertilization from the zygote, and it is resulting into the development of the adult tissue or the organs. So that there was the differentiation of the different cells, that is the egg cells and sperm cells, into the different parts only after the fertilization. This concept of that epigenesis was universally accepted. Sixth one is that theory of the acquired character. This theory was proposed by the famous French biologist Lamarck. According to this theory, once any character, a new character acquired by the individual, and that will be passed on to its progeny. So whatever that any new character, and that character, if it is acquired by an individual, and that character as it is passed on to its progeny. But it means that, for example, the man's develops a strong muscle by exercise. So all his children would have the strong muscles. If there is the on the another hand, if the person becomes the weak, and all the children from that person would have the weak, all the children's weak. So that is whatever the characters which are acquired by the individual, those pass on to its progeny. That is according to the theory of the acquired character. But this theory was disproved by the Weizmann. This theory was disproved by the Weizmann by conducting the one of the experiment on the mice. That he cut that tail of that mice, and mating in between the two mice, and whatever that offspring he got, having with that of the tail. So he repeated that experiment, that is for the 22 generations, but whatever the offspring develops still have the long tail. So that theory of acquired character was disproved by the Weizmann by this experiment. Seventh one, that is germplasm theory, and that theory was advocated by the August Weizmann, German biologist. According to this theory, the body tissues are of two types. That is, one is germplasm, and second one, that is somatoplasm. So that germplasm refers to that reproductive tissue or the cells which those are responsible to produce the male gamete or the female gamete. So that germplasm tissues are involved in the reproductions. So another one, the another body tissue, that is the somatoplasm. So that is other body tissues, and which are not related to the sexual reproductions. So according to the germplasm theory, there are the two types of the body tissue. One is that germplasm, and second one, that is somatoplasm. That germplasm, it is in relation with the reproduction, and the somatoplasm, it is not related with the sexual reproduction. So that the transmission of the characters from the one generation to the another generation, that is only through the germplasm, not through the somatoplasm. So if there is any change in the germplasm, will leads to change in the next generation, and that theory was accepted in broad sense. Then the theory of the Pancenes. This Pancenes theory was put forth by Charles Darwin. According to this theory, very small and exact, but the individual copies of each body organ, that called as a gamules, and that is small body organs which are already present in the bloodstream. According to this theory, these small body organs, very small body organs already present in the bloodstream, and those are transported from the bloodstream to the sex organs. And that gemmules which are assembled in the gametes. So whatever that gemmules, that is one gemmule for the one body organ, another for the another body organs, and all these gemmules which are present in the bloodstream, and through that bloodstream it is transported to the sex organ, and that all the gemmules which are having with that of the different organs, those are assembled in the gametes, and after the fertilizations, that gemmules moves into the different parts, that is the in the development of the respective organ, that is the development of the respective organ, and this is the gemmule for that particular organ, this is the another gemmule for that particular organ, and after the fertilization all the gemmules it is assembled, and then there is the development of the individuals. But if there is any gemmule, if it is defective, that is responsible for the development of the defective organ in that particular individuals. So this one was the theory of the Pancenes, and this theory was again rejected because of scientific base. They cannot prove that scientifically that the gemmules which are present in the bloodstream and those are assembled after the fertilization, and that each gemmule having with that of the each body organ, and it is assembled, and there is the development of the offspring. So it was again rejected. Then coming to that, this is all about the pre-Mendelian genetics, or that pre-Mendelian era. Whatever that Mendelian, what about that Mendelian era? That is the overview of the Mendelian genetics. Father of genetics, because of his work, there is the one new branch of biology, that is genetics. He Australian monk, and he conducted his experiment with the garden pea, and he thought that the heritable factors or genes retain their individuality generation after the generations. So this is the place where Mendel worked on the monastery garden. And what is the biography of the Mendel? Regarding Mendel, born in 22nd July 1882 in Silesia, Austria, graduated from the Vienna University in the year 1840. After the graduation, he returned to that Augustinian monastery and appointed as a monk in the year 1841. Then he worked as a substitute teacher, that is for the physics and the natural science from the 1854. Then in the year 1857, he started his experiment, that is the hybridization experiment on the garden pea. Whatever the work from that 1857 to 1864, about the 7 years experiments, span of that experiment, in the year 1864, he presented his result in the Natural History of the Society of the Brunn, and in 1866, he published his paper entitled that Experiments on the Plant Hybridization. Again, that in the year 1860, he was appointed as a monastery, and he had this post till death, and he was died in the year 1884, in 6 January 1884. So from that 1864, he presented his result in the Natural History of Society of Brunn. But from that 1864 till the 1900, whatever the work was done by Mendel, which was overlooked, that is about 36 or 37, 35, 36 years. So that work was again rediscovered by the 3 different scientists, by Karl Koerens in Germany, then the Teschmark in Australia, and Hugo de Vries in Holland. Individually, they found the same result as like of Mendel. So there is the rediscovery of Mendelian work in the year 1900. So now I will just tell about that. What was the Mendel experiment? That Mendel experiment, he made the cross in between the 2 plants. He selected the garden pea for his experiment, and he made the cross in between these 2 plants, and having with that of the contrasting characters. So whatever that, if 7 characters studied by Mendel, by chance all the characters studied by Mendel, it is qualitative characters. So on the crossing, Mendel found that after the crossing in between these two contrasting characters, for example, if it is the one plant, it is in tall condition, another plant, it is in short condition. After the crossing, he found that in the next generation, in the F1 generation, whole individuals obtain in F1 showing the only one trait, that is from the parent. It may be the tall or it may be the short. It is depends on that which character it is dominant over the another one. The character only in case of the F1, and that F1 it is in the heterozygous condition, and all the plants showing the one of the character from the parent, and that plant the whatever the character which is dominant, it is expressed in F1. So that they are showing the only one trait only in case of the F1 generation. In the next stage, that is selfing of that F1. He made the selfing of that F1 interbred, and Mendel found that F2 generation, that new F2 generation show the different results. He got the different results. If in case of the monohybrid, in dihybrid, he got that ratio it is 3:1 in case of the monohybrid. Every three individuals showed the similar traits of one parent. That is, he got the ratio, it is 3:1. It means that the 75% of the population it is having with that of the tall character, and 25% of the population it is having with that of the short character. It means that, so this one was the whatever the plant selected for the crossing, it is in the homozygous conditions. Pure lines are used for that crossing, and he got that F1 it is in the heterozygous conditions. So only in that F1 it is in the heterozygous condition, and F2 he got that all, that is as like of the parent as well as in the heterozygous conditions. So that dominance effect was observed by Mendel in his experiment. So on this experiment, he formulate that law of segregation and the law of independent assortment. I will not go through that in detail, but law of segregation, it means that when alleles for the two contrasting characters, so alleles from the two contrasting characters, when those are come together, if it is that tall character is there and the short character is there, and whatever the alleles for that, if it is that for the allele for the tall character it is T, and the allele for the short character it is small t. When these two alleles having with that of the two contrasting characters, those are came together in the hybrid, but they do not blend, they do not contaminate, they do not affect with each other. They will not affect on each other. They are separating from each other during the meiosis. They are separate from each other, and those separation of that alleles in the pure form, and those are passed on to the different gametes in the pure, that is in the hybrid. So that there is the gamete formation, that capital T and small t, those are segregated from each other, and that alleles again pass on to the different gametes, and there is the formation of the hybrid having with that of the capital T and small t. This is law of segregation. Second, that is law of independent assortment. According to this law of segregations, only one character it is involved, that is monohybrid cross is there. In case of the law of independent assortment, there is a dihybrid cross is there. He considered the two characters, how the characters it is inherited from the one generation to the another generations. Simultaneously study the two characters, so that in that case the two pairs of the genes which are involved in the crossing program, so that is dihybrid cross, and that two gene pairs it is entered in the F1 combination. When these two pairs from the two pair of genes enter in F1 combination, that both of them have the independent dominant effect, and these genes segregate, and there is the formation of the gametes form, and the assortment occurs randomly and quite freely. That is in case of the law of independent assortment. Here it is that two gene pairs it is entered in the F1 combination, and that both of them have that independent dominant effect, and these genes segregate. There is the segregation of the genes when there is a gamete formation, but that assortments occurs randomly and quite free. Each and every gene have an equal chance to combine with the another one. So this is in case of the law of independent assortment. These are the characters studied by Mendel: seed shape, seed color, flower position, pod color, pod shape, plant height, and etc. So he made that distinct classes. That is in the which one is that dominant, which one is that recessive. Luckily, whatever the characters studied by Mendel, all the qualitative characters. So in case of the plant height, tall and dwarf, he didn't measure in tape. That is by naked eye. This one is the tall and this one is the short. So whatever the characters studied by Mendel, all the quality characters are there. Then why the Mendel selected the pea plant? So success of Mendelian experiment was one of the selection of the plant, that is the pea plant, garden pea plant. Because of that distinct phenotypic characters, the one is that distinct phenotypic characters that particular plant having with that of the morphological characters. It may be the flower color, it may be the pod color, it may be the pod shape, and that can be observed, that can be detected by the naked eyes. So another one is that short duration crop, that short lifespan. The garden pea takes about 60 to 80 days to, that is, it is mature within that 60 to 80. So that plant, it is a very short lifespan. Then the third one, that is a highly self-pollinated crop. This garden pea is highly self-pollinated crop, and that, only because of that, that particularly purity, that purity can be maintained only because of the self-pollination. Then the bisexual flower, it is both the sexes which are present, male and female sexes which are present in that same flower. The size of the flower, it is big, so it is very easy for the hybridization, that is for the emasculation and the pollination. And another one is that, so whatever the hybrids develops after the crossing in between the pure homozygous lines, between the crossing in between these purely homozygous lines, he got that hybrids, it is in the 100% fertile, because whatever the genetic constitution of both the parents, it is complementary with each other. So this one, these are some reasons for that selection of the pea plant. Then the reasons for the Mendel success. Mendel's success, the first one is the choice of the parent. He selected the garden pea. It is a very short duration crop. It is self-pollinated crop, as I told you earlier. Second one is that attended the only one character at one time. He attended that, only he focused on only one character. At the time of the hybridization program, he focused on only one character, at how that character it is inherited from the one generation to the another generation. Third case is that he kept the record, or whatever that observation he made, all the records maintained in detail. That is the third reason for the Mendel success. Then the whatever the observations recorded by Mendel, that is observation recorded by Mendel, that is having with that of the distinct classes. He can make the classes. That is, he could make the classes in case of the tall and dwarf, then the axial, then in flower position it may be the axial, in case of the terminal. So he could divide that observation in the distinct classes. Then another thing is that he studied the large population, that is to avoid the error. That is from his experiment, about 28,000 plant population was observed and recorded the different characters on that particular population. So the huge population used for his study, that is to avoid the error. Then the another one is that as you know that Mendel was a very good mathematician. He is not a biologist. So he applied that mathematical knowledge, that is to interpret the result, and with the statistical method he could interpret that result in the ratio. And another case is that knowledge of the shortfalls of the earlier workers. He studied well. That whatever the shortfalls, why they earlier workers failed to conclude that how so there is the shortfall in, so whatever the knowledge of the shortfalls of the earlier workers, he found that he studied well, and it helps to Mendel to new directions. So these are some reasons for the Mendel's success. Another question is that why Mendel works remain in dark, that is from the 1866 to the 1900. That is scientists at that era, scientists believe only on the Darwin's book, Origin of Species, because whatever the results from Mendel was completely opposite to the Darwin theory. So in that era, the scientists only believe on the Darwin's book. Another case is that the scientists had a little knowledge of cytology and genetics, and the Mendel was not a biologist. He was a mathematician. So nobody is believe on his experiment because he was not a biologist. And another case, another thing is that Mendel did not advertise his work, because from 1866, 1864, he presented his paper. In 1866, he published his paper. From 1864 till the date of that, till his death, didn't advertise his work. So that Mendel work remained in dark from the 35 or 36 years up to the 1900s. Then coming to that post-Mendelian theories of heredity, it is already covered by me, the Gregor John Mendel, father of genetics. So whatever the principles, Mendelian principles, that all these principles again clarified by the different workers, several workers, and from that principles again confirmed by the several workers at the time of the clarification, at the time of the confirmation, some workers found the different reasons of the heredity were these new concepts were different from the So these new concepts, whatever the new concepts came forward after the Mendelian work, those are completely different from the findings of the Mendel. So that comes under the Mendelian deviations or the exceptions or the post-Mendelian concepts. So these are the era, that is the pre-Mendelian era, then the Mendelian era, and the post-Mendelian era. In that era, they got some results which were different from the findings of the Mendel. So we will see that one by one. These are some Mendelian deviations, that is incomplete dominance. Second one, that is co-dominance, multiple allele, linkage, lethal genes, gene interaction, pleiotropic gene effect, polygenes, environmental effect, and the cytoplasmic inheritance. Now, incomplete dominance. Mendel worked believe only on the complete dominance. Now that is as per that, if the red flower plant crossed with the white, he got that red, because that red it is dominant over white. So whatever the work whatever the work of Mendel, which was only related with the complete dominance. Now after that, some workers found that there is an incomplete dominance. Incomplete dominance, it means that when the red color flower cross with the white flower, the F1, it is not a red, not a white, but it is some intermediate effect, that may be the pink. So that red color, it is not completely dominant over the white. So in that case, they got that some intermediate effect, that is pink. So in case of the incomplete dominance, the phenotypic ratio, as well as whatever that as per monohybrid phenotypic ratio, as per Mendel, 3:1, that is the 3 red and the 1 white. But it is modified into 1:2, 2:1, that is only because of the incomplete dominance. And that in case of the incomplete dominance, both, that is the phenotypic ratio and the genotypic ratio, it is same, that is 1:2, 2:1. Then second one, that is co-dominance. That co-dominance, it means that when the alleles from the two different parents, those are combined together in F1, it is in the heterozygous conditions, both the alleles express their phenotypic effect when it is in the F1, and that is called as a co-dominance. In this image, you will observe that the chicks, it is having with that of the blue in color and the gray in color, and the whatever the F1 it is having with the both the colors, so that both the alleles will express in their phenotypic in the heterozygous condition. That is called as a codominance that was not observed by Mendel. So this image will give clear the idea about that complete dominance, codominance, and the incomplete dominance. In case of the complete dominance, if there is the blue cross with the yellow one, you will get that blue. It means that blue it is dominant over the yellow. Second one, that is codominance. When the blue it is crossed with the yellow one, you will get that both the alleles expressed in the F1. So you will get that combination of both the colors. And in case of the incomplete dominance, you will get the different one. When the blue cross with the yellow, it may be the green or another one. So in F1, it is the completely different from that. Then the multiple alleles. This is the another Mendelian deviation or the Mendelian exceptions. So the multiple allele, it means that Mendel worked only on that only one gene pair. It is involved in the crossing program. Only one gene pair. It is responsible for the expression of the characters and etc. So the gene mutation, it is responsible for to development of the many different alleles of the genes. So whatever that, if there is the more than two, if there is the number of genes more than two, that is called as a, that is, you know, that the allele is the alternative form of genes. If that number is more than two, then that is called as a multiple alleles. So any two of these can be present in the genome of the diploid organism, but the haploid or gamete can have just one of them in its genome. One thing is that the multiple alleles always occupy the same locus of the chromosome, as well as that multiple alleles always influence the same character. They are responsible to express the only one character. Another case is there is no crossing over takes place in between the member of the multiple allelic series. So this multiple, very good example of this multiple allele is the fur color in rabbit. There are the four types of the fur color, that is the wild type one, chinchilla one, then the Himalayan, and fourth one, that is albino. This is the one of the character, that is the fur color in rabbit or the coat color in rabbit, and that was governed by the one of the gene, that is capital C. But having with that particular gene, having with that of the series of the alleles, it may be the capital C, then the capital CCH, then the capital CH, and the small c. These are the series of the alleles, and those are responsible to express the different phenotypic appearance. So that is the multiple allelic series, and that multiple allelic series, it is responsible to express the only one character, that is coat color. All the multiple allelic series, it is present in the one locus, but that expression of that allele, it is different. It is at a phenotypic level. Another one is that linkage. What is that linkage? That was not observed. It was exactly opposite of the law of independent assortment. Mendel always observed that the independent assortment of the genes. It means that each and every gene assort independently, and it moves, that is assortment occurs randomly and quite freely, and each and every gene have the equal chance to combine with each other, and etc. So that linkage was not observed by Mendel, but later on the Batson and Punnett in sweet pea, then Hutchinson in maize, then by Morgan in Drosophila, observed that the genes having with the tendency to inherit in a group from the from the parents to the offspring. So that tendency of the genes, that is from the tendency of the genes, that is two or more genes, remain together in the same chromosome during the inheritance, that is called as a linkage. This is the one of the deviations. So in that case, if there is the chromosome is there, in that one chromosome, there is the two genes, and that two genes which are close, the distance in between the two genes, it is very less, and it is very close to each other, then chance of that linkage in between the two genes, it is very maximum. So that two genes could inherited from the parents to the offspring. If that genes, one gene it is present on the one chromosome, another gene if it is present on the another chromosome, the chances of the linkage, it is very rare. So there is no linkage in between them. Another case is, the third case is that if there is the gene 1, it is present, both the genes which are present on the same chromosome, but the distance in between the two genes, if it is very far away from each other, then in that case there is no linkage. So that linked genes, it means that whatever the genes which are located very close to each other, the chances of the linkage of that particular genes, it is maximum. So that genes, it is linked genes, those inherited together from the parents to the offspring. Another one is that lethal genes. That lethal means death of that particular organism. Some of the genes which are responsible to cause responsible for the death of that organism. That is, if that genes, if that particular organism carrying that genes, that organism will die. That gene, it is called as a lethal gene. Mendel findings were based on the equal survival of the all genotypes. So whatever that findings of Mendel, only based on that equal survival, whatever that F1, F2, all the individuality was survived. But in presence of the lethal gene, so whatever the normal segregation ratio, that is as per Mendelian segregation ratio, that is 3:1, and that ratio was modified into 2:1 ratio. How it is that? The one of the classical example of lethal gene, that is the coat color in mice. So the coat color in mice, whatever yellow coat color mice having with that of the heterozygous gene conditions, that is capital, for example, capital Y and small y. That when you made the cross in between these two yellow mice, and it is having with that the genetic constitution of both the mice, it is in the heterozygous condition, that is capital Y small y and the capital Y small y. You will get the F1, that is capital Y, capital Y, capital Y small y, capital Y small y, and small y small y. So that capital Y, it is also yellow. That capital Y, small y, it is also yellow. This capital Y, small y, it is also yellow. And small y, it is green. So, but here it is that the one of the gene that is in the homozygous dominant condition, that is capital Y, it is responsible to die that particular animal. So that the ratio, it is modified into 2:1, that is 2 LO and the 1. This is the classical example of the lethal gene. That lethal gene, when that gene, it is present in the homozygous dominant condition, and that gene, it is responsible for the dying of that particular animal. Another case is that gene interactions. In case of the gene interaction, the expression of the one gene depends on the presence or absence of the another gene. That is, whatever that number of genes, those are located on the chromosomes, and that expression of that particular gene, it may be depends on the presence or absence of the another gene, that is called as the gene interaction. So that in that case, another terminology related with that epistasis. So one allele at a one locus, that marks the expression of the allele which is present in the another locus, in the different locus. So there is the masking effect of that one allele completely marks the expression of the another allele, and they are showing the same phenotypic effect. That is called as a epistasis. And that gene which is responsible to marks the expression of the expression of that another allele, that another gene, that is called as epistatic gene, and whose expression is marked, that is called as a hypostatic genes. So the gene interaction, there are the different gene interactions with modification of normal F to phenotypic ratio, gene interactions without modification of the normal F to phenotypic ratio. The normal F to phenotypic ratio of as per the Mendelian segregation, that is 9:3:3:1. So the gene interaction is there without modification of the normal F to phenotypic ratio, and the gene interaction is there with modification of the normal F to phenotypic ratio, that is complementary factor, supplementary factor, and etc. Then the another deviation is that pleiotropic gene effect. That pleiotropic gene, it means that Mendel observed only one, that that one gene controls the only one character. One gene responsible for the expression of the only one character. But later on, the different workers found that not only the one gene it is responsible for the expression of only one character, but the number of one gene may be responsible to expression of the two or more characters. So that is the multiple traits. Maybe the single gene responsible for the control that 2, 3 characters, and that is called as a pleiotropic gene effect. Then the polygenes. As you know that there is the branch of the another branch of biology, that is the quantitative genetics. The Mendelian work, it is related with the qualitative genetic, qualitative characters, and in case of the quantitative, there is the polygenic characters are there. So that as per Mendelian work, which character it is controlled by the single gene, but later on the different workers found that some characters was governed by the several genes. So he put forth, that is the multiple factor hypothesis given by Nielsen. He observed that several genes which are responsible for the expression of the character, and each and every gene have the additive effect in the expression of the characters. So the one of the classical example is that the kernel color in wheat, the red color or the deep red, it is crossed with the white, and the medium red, he found that F1 it is medium red, and he got the when he observed that the ratio it is instead of the 9:3 as per the Mendelian segregation, the ratio should be the 9:3 3:1, but ratio it is modified into 15:1, that is 15:1, that is 15 red and 1 white. But that when that 15 progeny clearly observed, he found that there is a variation in the color. That is some of them it is in the deep red, some of them it is in the red, some of them it is in the medium red, some of them it is in the medium pale red, and etc. So that ratio it is modified into 1:2, 4:2, 6:2, 4:1 from the ratio of that the 9:2, 3:2, 3:1. Then the environmental effect. Mendel was not considered the environmental effect. Is there any environmental effect in the expression of expression of the phenotypic appearance? So there is some interactions in between them. That is the phenotypic expression. It is depends on the interactions in between the genotype and the environment. So that the importance of the environment in the expression of the phenotypic character, which was firstly realized by the Johnsons. So that environmental effect was not considered by Mendel. Is there any role of that environmental factors? It may be that genetic factors are there. In not environmental factors, it may be the temperature, humidity, and etc. Then the nutrients, chlorophylls. Those also affects on the expression of the phenotypic characters. Last one, that is cytoplasmic inheritance. That is, there are two types of the inheritance. One is that nuclear inheritance, and second one that is cytoplasmic inheritance. Mendel worked only related with the nuclear inheritance. So Mendel did not observe any differences in the reciprocal crosses. If there is the reciprocal cross was there, he didn't observe any differences in the reciprocal crosses or in the direct crosses. So the latter investigation revealed that there is the significant differences in the reciprocal crosses, and that leads to the concept of the cytoplasmic inheritance. So nuclear inheritance, whatever the genes which are located on the nucleus, those are responsible for transmission of the characters. In case of the cytoplasmic inheritance, whatever the cytoplasmic organelles, it may be the mitochondria or the chloroplast having with that of the DNA material, and those are responsible for the transmission of the characters. So whether that character it is from that nucleus, or that whatever the genes which are located in the nucleus, or which is from the cytoplasm, that can be detected by the reciprocal cross. So whether that character it is from the cytoplasm or from the nucleus. So that cytoplasmic effect, that is also known as maternal effect, or it is also called as extranuclear inheritance, or it is also called as a non-Mendelian inheritance, because Mendel worked only on the nuclear inheritance. he didn't found any significant differences in the reciprocal process. One of the example of the cytoplasmic inheritance that when the cross was made in between the two plants, having with that of the pale green leaved plant and the dark green leaved plant. That pale green plant, it is as a male, and the dark green leaved plant, it is as a female. So he got that F1, it is dark green. It is from the that is from the female parent. Again, he made the reciprocal cross. The reciprocal cross, the exchange of that is the interchange of the male and female parent. So that dark green, it was used as a dark green. It was used as a male, and the pale green it was used as a female. He found the different result. Instead of dark green, he found that pale green leaf. It means that the cytoplasmic effect is there when he got that F1 in that. He got that different results in the F1. So this is all about the chloroplast inheritance. And thank you very much.
Thank you, sir. Thank you for your detailed session. Then I request to Honorable Dr. B.J. Apparao, sir, Director, Collegiate Education, Mula Education Society, Sonai, Newasa, for presidential address.
Good evening, everyone. Today, really, I'm very happy to listen to the two stalwarts, Honorable K. Lavande sir, who remarked that the Mendel's laws will remain forever as long as the life exists on this earth. How correct it is. Similarly, he has taken us to the pre-Mendelian era, Hippocrates, Aristotle, to modern discovery of DNA, genes, etc. So I have no words to express my happiness and gratitude to Dr. Honorable K. Lavande sir. Similarly, we have another speaker, wonderful speaker, I say, Dr. M.G. Palshetkar, who has given very exhaustive lecture starting from pre-formation theory, means before the discovery of heredity, how the people used to think about heredity, pre-formation theory, theory of epigenesis, germplasm theory, etc. From there, he has given a full account up to the post-Mendelian period about incomplete dominance, codominance, linkage, cytoplasmic inheritance, whatnot. Everything is given. So really, we have really very good lectures today, and I thank these two guests for their wonderful contribution. Dear friends, we know Mendel has used a word called factor. Well, at that time, chromosomes were not known, DNA was not known, nothing. She has used the word factor. But as the science slowly progresses, advances, we came to know that factor is nothing but a gene. It is some sequence of DNA which can control the full character. So the genes now is equivalent to factor. Similarly, Mendel has used two terms, dominant gene and recessive gene. We know dominant gene is nothing but a gene which is the wild type. It is complete gene. No aberrations were there, no mutations were there. We call it as a dominant gene. The gene in which one or two nucleotides have mutated, or in short, a mutated gene is nothing but a recessive allele or recessive factor. So like that, if you go on nothing, even after hundreds, thousands of years also, whatever the Mendel, the great personality, has contributed to the science of heredity, that remain forever. I do agree with Dr. Lavande sir that Mendel's contribution will remain forever. And I appreciate all of you, all those who are taking keen interest in genetics, So we really, I'm sure you will all agree with me that we have two very good lectures, and there is, I think, no other better way to pay tribute to the great person, John Gregor Mendel. So I wish you all the best, and in future also your interest should be enhanced further by attending such lectures, and you will be contributing the science of genetics. Wish you all the best and thank you.
Yes, sir.
Thank you.
Thank you, sir. I request to invite to Mr. Gnaneshwar Tuvar, sir, Assistant Professor of Botany, Arts, Commerce and Science College, Sonai, for vote of thanks.
Good afternoon, everyone. I'm Gnaneshwar Tuvar, Assistant Professor, Department of Botany, Arts, Commerce, Science College, Sonai. It has been such an honor to be a part of this wonderful event. It gives me immense pleasure to deliver the vote of thanks on the occasion of national webinar on behalf of the Mula Education Society's Arts, Commerce, Science College, Sonai, and Yashwantrao Chavan College of Science, Karad. I would like to extend my heartfelt gratitude to the guest of honor, Honorable Professor Dr. K. Lavande sir, former Vice Chancellor Dr. Balasaheb Sawant, Konkan Krishi Vidyapith, Dapoli, who had highlighted the Mendel work. Thank you, sir. It takes immense pleasure in extending my sincere thanks to the guest speaker, Dr. M.G. Palshetkar, sir, Assistant Professor, Department of Agriculture, Botany, Dr. Balasaheb Sawant, Konkan Krishi Vidyapith, Dapoli, for describing the various pre and post Mendelian concepts very clearly to all the participants. Thank you, sir. My very much thanks to Honorable Dr. Bijay Apparao, sir, Director, Collegiate Education, Mula Education Society, Newasa, Ahmednagar, for his presidential address. Thank you, sir. I am thankful to Organizing Secretary Shushma Kirtane, ma'am, Department of Botany, Yashwantrao Chavan, College of Science, Karad, to organize such a wonderful webinar. Thank you, ma'am. My sincere thanks to the convener, Dr. Shankar Lavre, sir, Principal, Arts, Commerce and Science College, Sonai, and Chairman, Dr. S.B. Kengar, sir, Principal, Yashwantrao Chavan, College of Science, Karad. Thank you, sir. I am very much thankful to organizing team, all the faculties, technical team, for their continuous efforts to make this webinar a grand success. Thank you all. Last but not least, I am thankful to all the participants who made the event success. Thank you, one and all. Thank you.
Thank you very much on behalf of Arts, Commerce, Science College, Sonai, and Yashwantrao Chavan College of Science, Karad. Thank you, participants. Thank you, guest speaker and Honorable former Vice Chancellor Dr. K. Lavande and Dr. M.G. Pancetkar. Thank you.